2020. Please join me in welcoming Oleg Nodelman to the stage.
Thanks a lot. So my name is Oleg Nodelman. I'm the founder and managing director of EcoR1 Capital, you guys can come a little closer. A $1.5 billion Zoom and San Francisco-based biotechnology-focused investment fund. And I want to thank the folks at Sohn for all the work they do, including my co-chairs, Graham and Josh. This is an awesome first-of-its-kind event. Biotechnology is complicated, and I only have 6 minutes, so my goal was to create a talk that even my 9-year-old twin girls here would understand. Biotechnology is an unusual sector in which, more often than not, success is idiosyncratic. Just because a company is lucky enough to get one drug to market doesn't mean that will ever happen again. And unlike technologies where each discovery enables subsequent discoveries to be faster and cheaper than the last, like computers and Moore's Law, most biotechnology companies are just some smart men and women in a room hoping to get lucky. Investing in these companies is akin to playing the slots in Las Vegas, but without the free drinks. Today, I'm going to tell you about a company that's created very different odds for itself, RLAY Therapeutics. RLAY has transformed the drug discovery process by using computer simulation and artificial intelligence to understand protein motion and function. This platform, called Dynamo, has opened a new frontier in drug discovery. Not too long ago, if a patient was diagnosed with cancer, all doctors really knew about the disease was which organs were affected. Doctors would prescribe the patient a non-specific chemotherapy that had been developed decades ago, and why the patient got better or worse was a huge mystery. Once we cloned the human genome, our understanding of the underlying biology of disease began to improve. A few years later, we started sequencing the DNA of cancer cells and noticed that cancer cells had mutations that were different from healthy cells. Now we know that breast cancer, for example, is not the same disease in every patient, but can be caused by a number of different mutations. Companies like RLAY make precision medicines that go directly after these mutations. Targeted oncology companies have been able to command significant valuation premiums. For example, just last year, Loxo and Array transacted for 8 and 11 billion respectively. And each of these companies only really had one key asset and no platform to be able to replicate their success. Most targeted oncology companies spend years designing, developing, and integrating on a single molecule. They're like a fruit picker in an apple orchard, trying to reach for one apple at a time. RLAY, however, has built a platform that uses AI to map how proteins move. This gives them the ability to develop drugs that standard biotech companies can't create, and they can do so in a reproducible way. RLAY is like this harvester moving through the orchard quickly, picking up opportunities where others never had a chance. RLAY has a higher probability of success to get drugs to market than other biotech companies, and they can also replicate this success on a massive pipeline of targets. RLAY's collaboration with D. Shaw Research is an important moat surrounding its business. The exclusive partnership provides the company access to Anton 2, their proprietary supercomputer that was custom built for performing molecular dynamics simulations, a technique that calculates the forces between each atom and every other atom in a given system at discrete time points in order to model behavior over time. Other drug discovery approaches may use molecular dynamics, but they're limited to less than 1/100th of the time scale that RLAY can achieve. A 10 microsecond simulation of a 1 million atom benchmark, which requires 1 day of processing on the Anton 2, 271 days on conventional hardware. That means RLAY can simulate the motion of larger molecules in far shorter periods of time compared to conventional forms of computation. What all of that means is that RLAY doesn't work off of a still picture like the figure on the left, akin to making drugs based on an old black and white photograph. Instead, they are working off of a full motion simulation, more akin to being inside of an IMAX movie, helping RLAY discover novel chemistry not just faster than other biotech companies, but in a way that no one else can. And with big data, there's a virtuous cycle. The more data you have, the better you get. RLAY has only publicly described a small portion of their pipeline. Let me tell you about their 3 lead programs, all of which go after specific cancer-causing mutations. Cancer, unfortunately for us, is smart. When we hit a cancer-causing mutation with a drug, cancer cells can mutate to avoid being killed, like a game of whack-a-mole. A target called SHP2 is at the top of a pathway commonly used by cancer cells to survive. RLY 1971 binds SHP2, blocking this escape mechanism. An effective SHP2 inhibitor can generate over $8 billion in potential annual peak sales in the US. FGFR2 is a receptor that can be co-opted by cancer to fuel its growth. But there are several FGFR types which look almost identical if you're looking at a static black and white image. Blocking the wrong FGFRs causes toxicity. But because RLAY can see how these proteins move, they've discovered a pocket unique to FGFR2. We believe RLY 4008 could generate close to $2 billion in potential US annual peak sales. Finally, PI3K alpha is an enzyme governing another pathway frequently mutated by cancer. Similarly to the FGFR program, blocking PI3K alpha is associated with toxicities due to where past inhibitors have bound. So RLAY found a new site on the enzyme to target. This drug stands to generate over $5.5 billion in annual peak sales. Remember that beyond these three initially disclosed wholly owned programs, there are literally dozens of other targets and a number of other disease areas outside of oncology that Dynamo could be applied to. With over $15 billion in peak sales potential just from disclosed programs, even more from the confidential portfolio, several years of cash on the balance sheet and an experienced management team, RLAY is ready to execute and the company's potential value is limitless. Thanks, you guys. Go back to school.
Now, I think, Oleg, are you going to introduce?
Yeah, I'll go ahead and introduce Biwa. So Biwa Chen founded Cormorant in 2013. She's one of the most clever, hyper-intelligent, and bravest investors I've ever had the pleasure of working with. And in fact, I consider Biwa to be my retirement strategy if she'd ever open up her fund to let me invest in it. I can't wait to see what she presents so I can immediately buy some.
Thank you very much, Oleg, for such a kind introduction. Can you see the slide? This is the disclaimer. So I'm going to talk about a company called Forma Therapeutics. Current market cap is $1.7 billion. It has two product candidates. One is to treat sickle cell disease and one is for prostate cancer. So sickle cell disease, as many of you know, is an inherited disease that reduces life expectancy as well as hurt the life quality a lot. When a child inherits one good and one bad copy, he will live a normal life. When he gets two copies, that's when the trouble begins. So the hemoglobin of this specific type have tendency to stick together, polymerize, and cause the red blood cell to not flexibly change shape when it passes through the small blood vessel. And this condition itself, actually, because of the anemia, will upregulate a few molecules that actually make it even worse. So as you see on the right side of this graph, basically there's a molecule called T3DPG, and then later in this glucose metabolism, you use that to make ATP. So when 2,3-DPG got piled up, you not only have a molecule that now grab onto the make the oxygen release more easily to from the hemoglobin, because that's what the body trying to do to get oxygen. But as a result, the oxygen affinity change will make the cell even worse. So it's worse in multiple ways. That reduced ATP production is also very bad for the health of these red blood cells. So this is a natural regulatory pathway in the red blood cells. And people have cleverly tapped into this biology to activate PKR. So if you look at the history of biotech, there's a company called Agios that has already gone into clinical trial and validated this target. They have shown increased hemoglobin level in sickle cell and a beta thalassemia patient. But that drug has two problems. One, it accidentally inhibits an enzyme that alters the sex hormone level, so we don't want that side effect. Another problem is drugs are metabolized by our liver enzymes, and this molecule induces the enzyme that metabolizes this drug. So it's kind of a self-defeating circle that you can't get the blood level very high. So if you look to the left side of the slide, the good thing about FT4202 is it addresses both problems. So it can achieve consistent level of drug in plasma that reach optimal therapeutic level, and it has shown no risk of a sex hormone disturbance. The drug has gone into human. After one single dose, it has shown three things. It almost normalized the level of 2,3-DPG and ATP. Oxygen saturation curve now look almost normal. It also has altered the point of sickling for these red blood cells. And the third thing, in response to osmolarity pressure change, it shifts the curve in the right way. So a competitive product called I forgot the name, global blood drug. Basically, if you look at the third point, it shifts the wrong way. This is the first drug is actually shifting the right way. That detail is very important. So the STOK basically is going to look forward to the multiple dose result, which will be presented in December this year and the summer of next year. And Forma has a second drug candidate that's really interesting. So if you look at the right side of the slide, prostate cancer is mainly driven by androgen receptor, which bind to DNA and turn a lot of gene on that makes cancer cell grow really happily. But it cannot do it alone. It needs a lot of cofactors. One necessary cofactor in the darker green is called CBP-P300. When you take that one away or inhibit that, actually it will turn off the gene expression even with AR signal is there. So look to the left side, the application of this drug will be initially for patients who have failed and show resistance to the AR inhibitors, which are multi-billion-dollar drug right now. It's like that standard of care for this kind of patient. But 20 to 40 patients, they show resistance. And then if they prove it works alone in resistant setting, the natural thing to do next will be to combine with AR inhibitors upstream to make the period of time patients stay cancer-free or reduce cancer burden for a longer time than either drug alone. So we look at the preclinical data of this compound in various models. It has shown very good efficacy in mice implanted by with cancer that's resistant to current best drugs. And this drug is about to start clinical trial in fourth quarter of this year. So these trials take a little bit time to show sign of efficacy. My estimate is it could take up to a year around there to know. So in summary, I think that's a good risk reward stock because if either work out, it'll be bigger than current market cap. If both work out, it could be multiple fold from here. So I'll stop here and I thank you for the opportunity.
Thanks, Biwa. Thanks, Biwa. Next, we've got Aaron Davis. My good friend Aaron is the co-founder and CEO of Boxer Capital. the healthcare focus of the Tavistock Group. Boxer was founded in 2005 with $6 million in seed capital and has grown today to over $2 billion, with over 80% of that coming from performance. Aaron's based in San Diego, where he's often confused for Jason Statham. He runs a very concentrated portfolio, so all of his children are his favorites, and he's going to tell you about one of those today.
Well, thanks, Oleg. I appreciate that nod. So following BWA is never an easy task, but I'll give it my best shot. Thanks for having me here today, taking the time to listen about one of my children, as Oleg noted, Odinate Therapeutics. Odinate is a cancer drug company that is developing an innovative form of an already proven very active drug used currently for metastatic breast cancer. The drug's name is Tesitaxel, and it's an oral formulation of a taxane that was just shown to work successfully in its last phase of human studies before filing for potential FDA approval. The company's market cap is down 70% from its 52-week highs, sitting at a $600 million market value with $200 million in cash, enough runway to hopefully obtain FDA approval. They will be filing for this mid-next year. We are going to walk through three main controversies surrounding the stock today. Controversy 1, will this drug get FDA approved? Controversy 2, is this drug attractive to physicians? And controversy 3, will this drug be a commercial success? Before I go through these dilemmas in detail, though, first let me give a brief background on the next slide of the disease setting of metastatic breast cancer. Metastatic breast cancer unfortunately accounts for 271,000 new diagnoses per year in the U.S. and 42,000 deaths per year in the U.S. Once breast cancer becomes metastatic, it ultimately becomes a terminal disease. When treating metastatic breast cancer, it should be known that clinical benefit for the patient is a balance between efficacy of drugs, tolerability of drugs, and ultimately the quality of life. There are several treatments that patients cycle through over time, and chemotherapy remains a mainstay in the treatment paradigm. Taxane is a class of drugs that today is used approximately 40% of the time for first option chemotherapy and is administered as an IV infusion, causing significant hair loss and peripheral nerve pain. Another drug widely used in this setting is capecitabine, an oral treatment that unfortunately causes painful rash on the hands and feet alongside GI upset. Odinate's drug, Tesitaxel, is a new oral formulation of the current taxane standard of care, given 2 to 5 pills one time every 3 weeks without the hair loss and much less nerve pain associated with the IV infusion taxane. So now I'd like to cover controversy 1. Will Tesitaxel obtain FDA approval? The main takeaways I want to leave you with here are the following. The FDA design for their phase 3 trial is consistent with the FDA guidance, as well as many recent FDA approvals of other drugs in this setting. The efficacy demonstrated in the trial is meaningful to patients with terminal disease and consistent in magnitude of effect from other studies of FDA-approved drugs in this class. The patients enrolled in this study were of the proper baseline characteristics of prior treatment experience with taxane usage and other commonly used drugs in the U.S., being that a majority of the patients were enrolled in the U.S. as the FDA typically requires. Another main point to cover here with the data is on grade 3 neutropenia, or neutrophil reduction, which is a significant hang-up for some investors. Neutropenia is a very manageable phenomenon to the oncologist and is completely reversible, and we do not think that this will get in the way of an FDA approval for a oncology drug in the metastatic setting of breast cancer. So now I'd like to talk about, will this drug be attractive to physicians? Grade 3 neutropenia is, again, being focused on by many as impeding the potential attractiveness of this drug to physicians. Again, neutropenia, we believe, is reversible and manageable by oncologists in the clinical practice through dose adjustments and modifications. And it's completely reversible toxicity that is not painful to the patient and does not really impede efficacy for the patient. Toxicity associated with the Contessa study was consistent with what is expected from combination treatment using a taxane and capecitabine together. Usage of Tesitaxel monotherapy causes significantly less side effects, and neutropenia, as you can see, and still gives great efficacy as an option for physicians that may prefer to treat sequentially versus in combination as the trial was done. Taclitaxel is an oral formulation of the current IV standard of care, but does not cause hair loss and significantly less peripheral nerve pain, two of the most limiting side effects for many women battling this disease. This represents a significant driver to the patient's quality of life, alongside providing the current treatment utilized. So for these reasons, we think doctors will use taclitaxel if FDA approved. And last, I'd like to talk about, will taclitaxel be a commercial success? Taclitaxel capecitabine combination showed a statistically significant and clinically significant result, and if FDA approved, should lead to reimbursement as a combination therapy. The National Comprehensive Cancer Network, or NCCN, developed a recommended set of guidelines in the U.S. for oncologists to follow for the treatment of mostly all cancers. Drugs that are FDA approved usually find their way onto NCCN guidelines in multiple settings over time. This should enable physicians much more flexibility with how to utilize an orally administered alternative for women with breast cancer. So, in conclusion, I believe ODT represents a significantly undervalued company in the oncology sector, where FDA-approved assets that are wholly owned are incredibly scarce and valuable to larger companies, as you can see with some recent acquisitions in the breast cancer space like Immunomedics. Taclitaxel is a new drug option that is more effective, has greater quality of life with less hair loss and nerve pain, and gives doctors and patients more treatment flexibility than the standard of care IV administration. I believe taclitaxel has generated the data necessary for FDA approval and will sell well upon commercialization. So I hope in this 6 minutes I was able to convey some key understandings of why I think ODT is such an underappreciated.
It's the CIO of RTW Investments, where he and his team focus on investments involving innovative drug development. Rod founded the 4th largest gene therapy company in the world, which is called Rocket, and he closed the first biotech SPAC deal last year, the most successful to date, which may have helped give rise to the biotech SPAC trend this year. He serves as an adjunct associate professor at NYU Stern. Before founding RTW, he was the healthcare PM for Davidson Kempner. And before that, he worked for Wendy Holman's husband, Wayne, at Point 72. Rod, welcome to the stage.
Hey, thanks so much, Graham. It's great to see you. It's actually been a little while, so it's nice to see your face. Let me try to share my screen here. Okay, hopefully you guys see this deck that's up. Great. So it's great to be here, obviously in support of such a wonderful cause like the fight against pediatric cancer. I do have some pretty fond memories. When I was starting out as a PM about 15 years ago, I would listen to these pitches at the Lincoln Center events. And I think it's just really wonderful, right? That since that time to see science really move from the grant side, to also include investment ideas. I think it's reflective of the fact that despite that we're living in this really difficult time, we're also, right, indeed living in this age of incredible scientific innovation. So, I know myself personally, I feel really blessed, right, to have a career supporting medicines. So the example I have for you today is this company called STOK. So STOK is developing a RNA medicine for a disease called Dravet. And Dravet is a rare, truly tragic pediatric genetic cause of epilepsy. So these patients, they have seizures in their first year of life. They're generally refractory to treatment. There's a lot of mortality that goes along with this, and a significant proportion don't survive to becoming adults. But the vast majority that do survive they also have really significant morbidity. So they have cognitive impairment, they have motor control issues, and ultimately, for the bulk of patients, their seizures persist throughout their lives. So importantly, Dravet is a genetic disease. So 3/4 of patients are caused by a gene mutation in a gene called SCN1A. And this gene, with this gene mutation, it reduces the number of a specific type of sodium channel. It's called NaV1.1. And in essence, you can think of Dravet as a disease of missing channels, right? And these are the channels that are needed for proper electrical signaling in the brain. Okay, so I called this an RNA medicine. So what do I mean by that? So up until about a half dozen years ago, the major drug classes that have been available were small molecules and antibodies. Is the deck flipping? I'm not sure. Maybe Graham, if you unmute, can somebody tell me if the slides are progressing?
Yeah, the slides are not progressing.
Okay, yeah, that's what I thought. Someone just sent me a message. Let me see here.
There you go.
Okay, do you see these slides now?
Yes.
Okay, let me leave it this way. I went into presentation mode, but that didn't seem to work. So we can walk through it this way. Okay, so let's talk about RNA medicines as a drug class. So up until about half a dozen years ago, the major drug classes that I'm sure all of us are familiar with were small molecules and antibodies. And what these do is generally they're interacting with proteins, right? So either with the business end of those proteins to up or down regulate them or modulate their activity, or in the case of antibodies, usually binding the proteins and targeting them for destruction, right? So, but as we all know, proteins are really the end or the output of a process, right? That starts with DNA. So DNA is the beginning. Think of it as the unique repository that all of us have that defines each of us and of all living organisms, right? And RNA is the thing that sits in the middle. And those activated RNA messages really are the things that define the unique attributes of any specific cell or tissue, right? So RNA medicines are then basically interacting with these transcripts, right, in the center of RNA, and they can also up or down regulate or modulate protein expression. So the issue with this tech, right, is the maturation took years. The short version of the story is it basically involved chemistry to make them safe, to make them effective at entering cells when they're injected as drugs. So here on this slide, hopefully you'll see that Spinraza was really the first poster child of the success of the application of RNA to a neurologic disease. This was a drug discovered by Ionis, has been partnered and then developed by Biogen, and it treats patients with the disease called spinal muscular atrophy, SMA. Here, the gene mutation results in also insufficient production of a protein, in this case, the SMN protein. It's also a terrible pediatric disease. It's a neuromuscular disease. It results in progressive weakness, respiratory failure, and then ultimately patients also pass away. So Spinraza, like you'll hear in a second for STOK, was designed to upregulate the SMN protein. So here you see on this slide on the left a first early mouse experiment that showed increased protein production. Then on the center panel, this is an experiment or a clinical trial in patients. It resulted in improved performance in an assessment of motor function. And then on the far right panel, you see that then in a randomized registration study, it resulted in a dramatic improvement in survival of these patients. All right, so then in a similar way for STOK, STOK 001 interacts with mRNA to favor increased functional channel production of this NAV1.1 protein. So let's walk through the analogous data. So far, we only have animal data, so we'll walk through that. On the far left, in Dravet mice, you see an approximate doubling of this channel protein. And then in the middle panel, you'll see this dramatic decrease in these Dravet mice in the rate of seizures. And then on the far right, a really, really impressive change in the survival of these animals. Okay, so here's where the status of the program is now. They dosed the first Dravet patient just this past August. They'll be dose escalating for the next several months. Next year, we would expect to see initial data. Hopefully, we'll see a difference on seizures from when patients were coming into the study. If there's a very strong signal, we're hopeful that then a registration study could start the year after that. And just to frame the investment, so the market cap of the company right now is about $1.5 billion. They've got about $200 million in cash. So it's an enterprise value of about $1.3 billion. They have enough cash to last into 2023. So just to frame two success cases and then failure, we think, in a solid result where you'd see a clinically meaningful reduction in seizures, this should translate to a blockbuster medication. A company could be worth $4 to $5 billion in that case. If it's really a transformative result, a near cure, it could be a $2 billion plus drug, and then the company could be worth north of $8 billion. If it is a failure, then we would think that downside in a worst case scenario would be the cash at the time of the data, or roughly $120 million or so. So thanks so much for your attention, and I hope you guys enjoy the rest of the presentations.
Thanks so much, Rod. So our final fireside, and then we'll have one more tout from Peter Coach.